All right, I think we'll start. So, yeah. Hi, everyone. Thank you once again for joining. I'm Judging, and I'm from SynBio.Oxford, and we are a student-run society that aims to provide educational and outreach platform for synthetic biology in Oxford. We do so by bringing you talks by experts from both academia and industry so you can learn from their knowledge and experience. And all this is thanks to our sponsor, Integrated DNA Technologies, or IDT. So today's webinar is titled Reprogramming Plant Root Growth Using Synthetic Developmental Regulation. And we are very excited to have Dr. Jenn Brophy with us. Jenn will be an assistant professor of bioengineering at Stanford starting September 2021, and her laboratory will focus on using synthetic biology approaches to engineer plants' form and function. Prior to this, she did her undergraduate in UC Berkeley in bioengineering, completed her PhD from MIT, and is currently working in Stanford for her postdoctoral research. As a recipient of multiple awards, such as the National Science Foundation Graduate Research Fellowship, she is truly an outstanding scientist. So the webinar today will be recorded, and please note that there will also be some time after the talk for questions. So if you have any questions during the talk, feel free to type it into the chat box and we'll address them after. So that's enough for me. I'll pass it over to you, Dr. Brophy.
Thank you. Okay, so let me just present this. All right, so I am interested in engineering plant form and function. So I'm interested in engineering the size and shape of plants because in plants, like in all biological systems, size and shape are important determinants of function. And if we can figure out how to engineer these features, I think we can create new and useful biological systems. On this first slide, I'm showing two examples in which humans have already changed the shapes of plants to produce something useful. On the top is modern maize, which was bred from its wild ancestor teosinte to increase both the size and the number of seeds the plant produces. This change in form has made corn into one of the most productive food crops we have today. On the bottom is a dwarf wheat variety, which was bred from its wild ancestor to decrease its stature so that you could apply massive amounts of fertilizer to the field and get an increase in grain yield instead of just more straw biomass overall. Now, I think these changes in form are super important, but I'm interested in engineering plant form, not just to make a larger corn variety, but because form is really important for environmental stress tolerance. So the shape and angle of a plant's leaves affect photosynthetic efficiency and the ability of plants to tolerate heat stress. The structure of the root system enables the plant to acquire water and essential nutrients from the soil. And even substructures, like these air channels that you can see in a cross section of a wetland plant, affect a plant's ability to survive in a challenging environment. In this case, the air channels enable plants to survive in flooded soils, where usually the flooding would submerge the root system and make it become hypoxic. These air channels deliver oxygen to the roots so that they can survive. I think the engineering of plants for environmental stress tolerance is going to become extremely important as global climate change alters the agricultural conditions in which we grow the plants that we rely on for food and also fuels, fiber, and medicines. These are just a few examples in the last couple of years in which different weather events have had catastrophic effects on the crop yields in the United States. Okay, so I, as a postdoc, have been focused on engineering root structure. And you may be wondering, like, why would you start with root structure? Well, there's a bunch of reasons, and I just want to lay them out for you really quickly. First, the developmental pathways that govern root growth are exceptionally well defined. This means that I have an idea of what genes I would need to tune up and tune down the expression of, and in what tissues, in order to have a specific effect on root structure. Then second, roots are tolerant to pretty extreme mutations. Here you can see some Arabidopsis plants that I tried to engineer that completely failed to initiate a root system, but the plants themselves still germinated, which means that I can get some information about what is happening in these roots and how to refine my designs so that I can actually build the root system that I'm interested in. So this tolerance to extreme mutations is extremely important. Third, plants undergo continuous de novo organogenesis for most of their organs. This is true of the root system. So along a single plant, you can actually look at many different time points if you were to think of time in terms of development. So you can see the beginnings of a root and fully mature roots all in one plant, which simplifies the analysis of your engineered system. A This, in contrast to maybe a mammalian system where you were trying to engineer organ structure in a mouse or something, would require you to sacrifice the mouse at whatever time points you were interested in and have many, many mice in order to look at kind of progression of organ development. So this feature of plants is useful. Finally, there's very simple cell lineage analysis because there's no cell migration in plants. So a cell stays put and you can actually follow its trajectory in time very easily. And finally, the structure of the root system is really important for the function or survival of the plant. So right away, if we can engineer root structure, we can have an impact on the plant's ability to survive in a challenging environment. Okay, so I want to engineer 3 different aspects of root structure, and I'd like to engineer each of these aspects independently so that these features can be tuned up and tuned down to create very diverse root systems. On this slide, I'm showing 3 simulations where we've increased or decreased either the branching rate, the growth rate, or the branching angle of a root system. So if you were to increase the branching rate, you would get more lateral roots per unit of primary root growth. If you were to change the growth rate, you might change how large the root system is after a given period of time. And the angle is the way that the roots grow with respect to gravity, these lateral roots, once they've emerged. So we can tune these independently. Then we may be able to create root systems that are optimized for growth in specific environments. These are 2 examples in which you may be growing a plant that's optimized for growth in a phosphorus limited environment. So phosphorus tends to reside in its bioavailable form in the upper levels of soil. So if you were to be growing in an environment where phosphorus was limited, you may want to have a root system that's very short and dense in that upper level of soil so that you can scavenge as much phosphorus from the environment as possible. In contrast, in a water limited environment, you may want to have a very long root system where the plants are reaching for water that might be in deeper levels of soil that haven't dried out as quickly as the upper levels might during a drought. So if we can tune these different aspects, we may be able to build these plants with really diverse root structures. Then the question becomes, how do you engineer root structure? As it turns out, engineering structure in general requires very precise control over gene expression. I'm going to illustrate this with Arabidopsis, which is sort of like the E. coli of the plant world. This is the easiest plant to grow and transform, or at least the most well-studied, so it has the most tools. So what I'm showing you here is Arabidopsis growing on a agar plate. And it's growing on this plate so that you can see its root structure. And what I want you to notice about the Arabidopsis root system is that it has a long primary root that grows down in the direction of gravity. It's got many lateral root branches, which also grow down in the direction of gravity. If you were to zoom in on this root system, you would see that it's covered in tiny root hairs. And these root hairs are important for water and nutrient acquisition. Now, if you make a single base pair change in this plant that modifies a developmental regulator, can have a really stark impact on root growth or the structure of the plant. So this single base pair change modifies the amino acid sequence of this developmental regulator, and it is expressed in a whole bunch of different tissue types. So the modified protein impacts many different tissues' development. Most strikingly, this mutation eliminates root branching. So you can see that there's no lateral roots on this plant. It also affects the plant's ability to sense gravity, which you can tell because the root no longer grows straight down. It's also a little bit shorter and it's no longer covered in root hairs. So just by expressing this mutant protein in all these different tissues, you have a bunch of effects on the plant's development. And its shoot system, you can see, also has several morphological changes. Now, if instead of expressing this mutant protein in all of these different cell types, if you limit the expression to a single cell type, you can make a targeted change in root structure. So here, the engineered plant does not have root branches, which is the targeted change we were trying to make, but it is otherwise identical to the wild type plant on the left. So our strategy for engineering root structure is to use very precise spatial control over gene expression to modify root development. Now, how do you do that? So there are a number of ways in which you can express genes in specific cell types. And one of the methods that you can use is to look through the literature for a promoter that has been well characterized and has the expression pattern that you're interested in. We're fortunate in Arabidopsis that there have been many different promoters that have been well characterized in this manner. So this is just an example showing you fluorescent protein expression in being driven by different promoters, and you can see which root cells those promoters are active in. But if there's no promoter in the literature that has the expression pattern that you're interested in, then you need to start being a little bit more creative. One thing you might think of doing is to mine promoters from an RNA-seq data set. So in this case, you would dissect out different cell types and then look for genes that are expressed only in the cell types that you're interested in, and then try to use the promoter region for that gene to drive your gene of interest. That sounds straightforward, but it's actually somewhat challenging to implement because we don't know what regions of the genome actually serve as the promoter for most of these genes. You might think that you could just grab, excuse me, the 5' genomic sequence for a gene and use that as the promoter. This paper has a nice illustration of why that can be challenging or why that may not work. So if you were to just grab the 5' genomic sequence of this ARF7 gene and use it to express a fluorescent protein, you'd actually see no expression in the root tip of Arabidopsis. Now that's surprising because we know that this ARF7 gene is expressed there. So the authors of this paper grabbed not just the 5' genomic sequence, but also the first introns and exon. And when they did that and fused it to a fluorescent protein, used this whole region as the promoter, they then recovered the expression pattern that they were expecting. Now, this is because there's information outside of just that 5' UTR that is important for defining a promoter's activity. So it's a little bit more difficult to just grab promoters from RNA-seq data sets than you might think. Okay, so these are the challenges. My solution to this is to use logic gates to try to control spatial patterns of gene expression. So in this way, we can take a few promoters that have known expression patterns and combine their activities in ways that give us new expression patterns in the plant. So here we've got 2 promoters. Just to illustrate how this would work, we've got 2 promoters. There's promoter A, which has expression pattern in the root tip. This is a cartoon of a plant root tip, and the green is supposed to show you sort of pseudo-fluorescent protein expression. So here you've got expression in just the root tip being driven by promoter A, and promoter B has expression in a subset of the root tip, which is a region of the plant called the columella, and expression in some of the central vascular cells. If you were to combine the activity of these 2 promoters using an AND gate, for example, you would get expression only where promoter A and promoter B are active, which would be those specialized columella cells in the tip of the plant root. So in this way, you can build a new expression pattern out of promoters that you have already characterized. In addition, if you can build genetic logic gates, you can integrate environmental signals. So you may be able to do something like respond to the presence of a fertilizer or other chemical in just specific cell types using an AND gate, and in that way initiate growth or a specific change in structure in response to an environmental cue. Okay, so that's why we want to use logic gates. Now, how do we build them? So in order to build logic gates in plants, I've created a collection of synthetic transcription factors. So these are synthetic activator proteins or synthetic repressor proteins we can use to build logic gates. The synthetic activators are composed of a DNA binding protein, which recognizes a very specific sequence of DNA fused to an activation domain and a nuclear localization signal. This protein, when it gets into the plant nucleus, will recruit RNA polymerase and initiate expression of any downstream gene. In contrast, the repressor proteins are just the DNA binding protein fused to a nuclear localization signal. And when that binding sequence is located in the middle of a promoter that would otherwise recruit RNA polymerase, it actually blocks the assembly of the transcriptional machinery and prevents transcription of the downstream gene. Now, for the DNA binding protein region of these transcription factors, I've used bacterial DNA binding proteins that have known DNA binding specificity. Now, there's a really large collection of them, which enables us to build there's a large collection of well-characterized bacterial DNA binding proteins. And using these allows us to build a large collection of plant synthetic transcription factors and a collection of promoters that are responsive to these transcription factors. So what I'm showing you here is just a synthetic activator and then the design of a promoter which responds to that synthetic transcription factor. And these little squares are supposed to be the sequences of DNA that these DNA binding proteins recognize and bind to. Okay, so these are sort of the goals Now I'm going to show you how we actually assay to see whether or not these synthetic transcription factors are functional in plants. So we use a Nicotiana transient expression assay to test to see whether or not these things are functional. And this assay is something that I think is super cool. And when I transitioned from non-plant work into plant work in my postdoc, thought this was wild that it worked. So this assay involves taking a plasmid that encodes your synthetic transcriptional activator and the promoter that's supposed to respond to that activator driving expression of GFP. You take that plasmid and you transform it into a special strain of bacteria called Agrobacterium tumefacens. You then infiltrate it into plant leaves. What that means is that you actually take a syringe and you press it onto the bottom side of the leaf and fill the airspace of that leaf with your solution of bacteria. The bacteria, once it's in the leaf, will then conjugate this plasmid DNA into any plant cell that it comes into contact with. The DNA, once it's inside a plant cell, will be expressed by the plant. So we can do experiments where we infiltrate a control, which is just the synthetic promoter driving expression of GFP, but without the synthetic activator to turn it on. We can infiltrate that on one side of the leaf. And then on the other side of the leaf, we can infiltrate a strain that has the synthetic activator in addition to those other parts. If we do this, we can see that we get really nice GFP expression only when the synthetic activator is present, which means it is turning on the synthetic promoter that we've built. Now, in addition to this GFP, we've actually got a constitutively expressed M-cherry on all of these plasmids. This just allows us to normalize GFP expression because there's some variability in how efficiently these agrobacterium conjugate DNA to the plant cells. And we want to control for that to make sure any variation in GFP expression we're seeing is actually due to the activity of our synthetic promoters and activators and not due to some other phenomena. Okay, so once we have these leaves, we can actually take hole punches and put them into a 96-well plate and use a plate reader to quantify gene expression. And for the duration of this talk, what I'm actually going to be reporting is this ratio of GFP to M-cherry so that we can kind of normalize out that noise we talked about before. Okay, so you can actually test, I've built 10 different synthetic activators and 10 different synthetic promoters, which these should be turned on by the synthetic activators, and we can test their activity in Nicotiana. What you can see is that for the most part, our synthetic activators are able to turn on expression of the synthetic promoters, and they are specific. So the synthetic activators are only turning on expression at the synthetic promoters that they were designed to turn on, not others. This isn't true across the board, but there is a subset of them, for which this is true. Now, for some of these synthetic activators, there's not very high gene expression. And what we can do, because these parts are synthetic, is just modify the activator or the transcription factor itself in order to make it function better. So in this case, what I'm showing you is the original design for the synthetic activator, which has VP16 as its activation domain. VP16 is actually a domain from a human herpes virus, and it functions okay as an activator in plants. But if we actually swap that activation domain for ERF2, which is a region of a plant transcription factor instead, you can see that we get much better activation. So although we've characterized them all with VP16, there is flexibility here where we can go in and tune the design of the synthetic activators in order to make them perform the way that we want. Okay, so this is the region of this grid where the synthetic activators are very specific. So these are able to turn on expression of just the promoters that they were designed to and not others in the set, which makes them the most useful for building synthetic circuits where we want promoters and transcription factors to be responsive to specific signals. Okay, so we can do the same thing with synthetic repressors and using that assay to look for their activity in plants. We can see that there are some synthetic repressors that function very well. So sorry, this is a similar graph to what I showed on the previous slides, but here we're only looking at the activity of these synthetic repressible promoters in the absence of a repressor. That's this control line along the top. And then the ability of the repressor to repress that promoter. We're not looking at the off-target effects because we assume that the off-target effects for the repressors will be the same as for the activators because we're actually using the same DNA binding proteins. Okay, so in some cases, this works very well. The repressor is able to turn off it's the synthetic promoter. But in other cases, the addition of the repressor doesn't seem to have much of an effect. And we think that this might be due to the mechanism by which we're asking these repressors to turn off gene expression. So if you remember, the repressor protein was just supposed to bind at the promoter region and interfere with the assembly of the transcriptional machinery at that promoter. But if this repressor doesn't bind very well, that activity is limited. So what we can do is actually add different repressive domains to our synthetic repressor and get much better repression in planta. So once we've got a set of transcriptional activators and repressors that we can use to modify gene expression in plants, how do we make them into logic gates? Okay. Well, I'm going to show you the design of a biological AND gate to give you a flavor of how we build these things. So on the left is a truth table, which is just going to show you for 2 different inputs, whether or not we expect to see output gene expression. So we have, in this case, no input, so neither input A nor input B are present. And we have our output gene, which is GFP, being repressed by a constitutively active repressor. So the GFP is being driven by, or would be driven by the synthetic promoter, but this repressor is being expressed and is turning it off. So our input A is an activator that would turn on expression of our output gene, but this repressor is epistatic to the presence of this activator, so the output is off. Our input B is actually a repressor which turns off expression of the first repressor, but because there's no activator present in the system, this GFP output is still off. And it's only when you have input A and input B present in the system that you get expression of your output gene. We can test to see whether or not these things are working with these same infiltration assays. In this case, we're going to be infiltrating 4 different spots on a single leaf. In the first spot is just the output plasmid. In the second spot is 2 different strains of agrobacterium, one carrying the output plasmid and one carrying input A. The second spot is just input B, and then in the last one, you have both input A and input B. You can see when you do these infiltrations is that it's only when you have both input A and input B present in the system that you get expression of GFP. Down here is the quantified fluorescence of these sleeves. And what I want you to notice is that in the cases where gene expression should be off to implement the correct logic, the bars are colored black. And in the place where we should see expressions colored green. I'm telling you this because on the next slide, I'm going to show a bunch of logic gates, and I want you to be able to quickly look to see whether or not these logic gates are functioning as expected. So, right, we can build not just AND gates, but a number of different logic gates in plants, or at least in the Nicotiana leaves. Now, Nicotiana leaf logic gates are cool, but they're not getting us any closer toward that goal of building new patterns of gene expression in whole plants. So instead of just doing infiltration assays in leaves, what we're doing is combining 2 different promoters, which have specific expression patterns in Arabidopsis roots, with all 16 different 2-input logic gates. And along the bottom here, you're seeing the different expected patterns of gene expression that you would get if you were to use these different logic gates to combine the activities of these 2 promoters. And you can see kind of a big variety of expression patterns when you apply different logic gates to these 2 promoters. I'm going to start by showing you real data of just these 2 very simple logic gates in plants. So these are called buffer gates, and they just recapitulate the activity of the input promoter. Okay, so our input promoter, A, is actually this sombrero promoter, and the control is just that promoter driving expression of GFP, and you can see nice expression in the root cap tip of Arabidopsis roots. Now the gate is going to have that same promoter A driving expression of a synthetic activator, which in turn drives expression of GFP. You can see when you do this, you get really nice recapitulation of the expression pattern of A. So this gate works well. Now that doesn't hold for every promoter. If instead you use a different promoter, so this is a pin-4 promoter instead, as your input promoter, you get an expanded expression profile relative to the control, right? So the addition of the synthetic activator in the middle is changing the expression pattern in plants. Now, why is that? Now, if you think about a multicellular system, tissue-specific promoters are actually what we should be calling tissue enriched. Now, the reason something like this may be happening is because the observed expression pattern that you see, for example, with that pin-4 promoter, is just the expression in cells where you are above a threshold of detection. So here in these columella cells and the central vascular cells, expression of the pin-4 promoter is very strong. And so using this fluorescent protein, you can actually see those tissue types lighting up. But if you were to maybe dig in a little deeper, you might find that there's a small amount of expression in other cell types. It's just below the limit of detection. And our synthetic system is actually, or at least we thought, amplifying expression in these other cell types so that they're now becoming visible. We tested this, and it is indeed the case that these synthetic buffer systems are actually amplifying signal. We know that because if you take a single promoter, this is a kind of weak constitutive promoter, and drive expression of GFP in those Nicotiana leaf assays, you see a small amount of GFP expression. But if you take that same promoter and you use it to drive expression of an activator that then drives expression of GFP, you see a massive increase in GFP expression. So it's serving as an amplifier and highlighting up these cell types in which we didn't previously see expression. Now that's kind of a problem if what we're trying to do is refine spatial patterns. So how do we tune up or tune down expression in these cells? Now, one way of doing this is to modify that activation domain like we talked about before. But another way is actually to modify the synthetic promoter. So in this case, we're reducing the number of binding sites for the activator and making the synthetic promoter less responsive to the amount of activator that's present. And here you can see the effect of having different number of binding sites in the synthetic promoter. You can actually get a nice gradient of gene expression where you have both more expression than just the control, which is directly driving expression of GFP, or less expression than that. So we should be able to tune up and tune down. I wanted to try to apply that ability to tune up and tune down gene expression to these plants that were lateral rootless, right? We know that by expressing this transcription factor in specific cell types in this plant, we can eliminate root branching. But a plant with only one root, just this primary root, is not as useful as one where we can actually tune the number of lateral roots. So what if we were able to control the expression levels of this developmental regulator and get a gradient in root branching, right? You might think that with the most expression of this mutant developmental regulator, we get no branching, but there's some sort of gradient in which we see fewer roots, or sorry, more roots being in correlation with the reduced expression of the regulator. Or we may find that there's a threshold above which expression of this developmental regulator no longer blocks root development, and then we get a bunch of roots being expressed. So either way, it'd be interesting to know, okay, how much of this developmental regulator do we need to actually have an impact on root structure? So we tried transforming different synthetic activators driving expression of this developmental regulator into plants. And what we saw was that even at our lowest level of expression of this developmental regulator, one where by confocal microscopy, we can't detect the expression of this, GFP fused developmental regulator, we still are impacting root development. So it seems like a really, really small amount of developmental regulator is enough to totally turn off root branching. And we may, in this case, want to operate even further down this expression control gradient. What I'm showing here is actually a completely uninfiltrated control. And you can see that there's actually some space in between the uninfiltrated control and our weakest synthetic promoter, in which we can build systems where we're driving an even smaller amount of expression. And those even weaker promoters might be useful for actually tuning development. Okay, so I've shown you two gates where things are, one works pretty well and things are kind of shaky with the other, I want to highlight a slightly more complicated gate because I think that these gates in particular are super exciting. These are what are called NOT implies gates. So NOT implies gates are on only when one of your inputs is present in the system. As soon as the other input is present, the gate shuts back off. So you can use this type of NIMPLY or NOT implies logic gate in order to refine spatial patterns of gene expression. So here you're taking expression at the root tip and you are subtracting out this second promoter's activity so that you get only expression in these little lateral root cap cells. Now, why would you want expression in just those? Well, aside from being able to just demonstrate that we can refine these spatial patterns of gene expression, we're interested in these cells in particular because of how important they are for gravity sensing in plant roots. So I mentioned before how we wanted to be able to tune the branching angle, but tuning this angle is actually all about tuning the plant's ability to, or the plant's response to gravity. The more strongly it responds, the steeper the lateral roots are that grow from this plant. Now, how does this gravity sensing work? Well, in those specialized columella cells, you actually have these starch granules, which when the gravity vector is applied to them will fall towards the bottom of the cell. Now, if these fall because your root tip is now oriented horizontally, then you get pulling on actin filaments in the plant and transport of auxin preferentially towards the side of the root that is like lowest in terms of gravity. And the addition of auxin on this side actually prohibits growth of the cells, and you get more growth along the top side of the root, which then causes bending. That sort of asymmetrical distribution causes bending. So what we can do with our synthetic system is potentially just express an auxin kind of signaling blocker in just the cells that would otherwise be responding to or doing that differential growth, right? And see if we can affect just root growth. Okay. So what I'm showing on this slide is a slightly modified version of what I've been showing before. And I'm using, in this case, this kind of brown color to denote expression of our developmental regulator instead of green. And in a wild type plant, what we see when we do this is that the plant root goes straight down. This is a much younger plant, so it hasn't started producing lateral roots yet. But when we express a developmental regulator in all of these root cap cells, we get a plant that does not sense gravity. It also severely messes with the development of the root cap and the differentiation of those cells. And what we're trying to figure out now, we have some preliminary results, but I don't feel confident in them enough to share today. But when we express just in these kind of other cell types, we can alleviate, we think, this weird developmental phenotype. And now the question is, what is the impact of the plant on the plant as a whole in terms of its ability to sense gravity? Okay, so that's everything that I wanted to show you guys today. I'm very grateful to my postdoc lab for supporting this work, and I am opening up my own lab in September, and I'm going to be looking for students and postdocs. So if you're interested, either reach out to me or apply to the Stanford Bioengineering program.
That's amazing. Thank you so much for that really interesting and clear talk. I really enjoyed it. So now we have time for some questions. So again, you can either raise your hand and unmute yourself to ask a question, or you can type your questions in the chat box. So while waiting for, oh, we have a question. Okay. So Renzi asks, do positional effects, the site of gene construct insertion, influence the activity of synthetic constructs?
Yes, absolutely. So in plants, genes integrate randomly into the genome when you do transformation. And the way that people often circumvent the variability that arises from different insertion sites is to average over many different plant lines. And so you kind of can alleviate some of the effects of this insertion frequency or the insertion bias. Yeah, so okay, that's one of the big challenges in this work, because then it requires looking at many different independent lines And what we're trying to do is be really good about reporting sort of the overall impact of the circuit. In the future, I would love to have just an insertion specific site or a location where we could put these transgenes every time so that we didn't have that problem. But I guess what I would like to say is that the overall impact of the circuit seems to override this insertion sequence bias. So while you do see some differences from plant to plant on maybe how strongly the fluorescent protein is expressed, or some tinkering around the edges in terms of which cell types may light up, you do see that the overall patterns are consistent across these lines, at least for the most part.
Thank you. Next up, we have Tony.
Yeah, so hi, Jen. Hi, nice to see you again.
Hi, nice to see you too.
So thank you very much. That was really a wonderful presentation. Very, very interesting. And I'm very happy that you start your own lab and everything is going great. I have a question that is a little bit related to this question that was raised. And I was wondering, because since you have a number of different repeats and you change that, how stable are these constructs in like across generations in Arabidopsis? Is that I know that can be a problem for these repeats. I wonder if this is stable or if you're running into issues, I guess also with multiple different alleles that you have to insert.
Yeah, it's a great question. So cross-generationally, I don't know. We've analyzed most of these things in the T1 generation, and I have T2 seeds all collected, but they haven't been analyzed yet. So I couldn't tell you whether or not the expression patterns hold across generations.
Okay.
What I can say is that when we started this, I was reusing a bunch of promoter pieces and terminator pieces across these circuits, right? So like all of them have a miniature 35S and the same terminator, and I was seeing a ton of silencing. I think because you would not get the expression patterns that you were expecting. So by swapping out some of these regions, we've been able to, at least in the first generation, not trigger silencing and see the expression patterns the way we had expected to see them. But it's a really good point that these things do need to be stable cross-generationally in order for these things to be most useful.
Do you think, like now you said the terminators, do you think would it be useful to use the natural terminators actually, like from the promoters that you use, and then also use the terminator region? Or is that, I mean, because that is basically another artificial point that you insert, right? I mean, and in principle, whatever there is a promoter region, there should also be a terminator region for that gene in the plant, right? So do you think that would be helpful?
Yeah, I actually don't know mechanistically how that would affect expression. It's possible that that could alleviate some triggering of a plant recognizing there's foreign DNA, but I have no idea whether that would work out. The strategy I've been using is just to take what have been characterized as known terminator regions for plants and make sure I have enough different versions of those so that I'm not repeating things in a single design. But it's a good point. That could be a good way to go about eliminating issues like this.
Okay, thank you so much.
That's great. Next up, we have questions from Francesco and Renzi, asking if you have considered using the CRISPR-Cas9 technology either to using the dCas9 to target like a specific sequence using CRISPR-A or CRISPR-I, or like using the TSKO system for refining the tissue expression boundaries.
Yeah, I think the TSKO system is super cool, and also like CRISPR-I, CRISPR-A have obviously had huge impacts in synthetic biology. I would like to use these systems as outputs of our synthetic logic gates to regulate expression of the developmental regulators that the plant is already has encoded in its genome, basically. But using them to build the synthetic circuitry, I think, is sort of a waste of using them, just because they're limited in how many targets you can probably activate or repress at any given time, right? If you have Cas9 fused to an activator, it's difficult to use it as a repressor. There's ways to get around that because there's multiple Cas9 varieties, and there's also like guide RNA handles that can recruit activation or repression domains. So you can kind of use Cas9 as both an activator and repressor at the same time, which is important for these circuits because you need both activation and repression for them to function. But I think their strength is really in binding whatever region you want in the genome. And for these synthetic logic gates, using a synthetic promoter that we've totally designed to like turn on and off expression of another synthetic regulator is totally sufficient. So I'd like to save using Cas9 for sort of the end output of these circuits, if that makes sense. But that's just my own personal preference, and I think that there is value in building out synthetic circuits using CRISPR to actually do the computation.
Nice, thank you. Next up, we have Aram who has a question. Aram, could you please unmute?
Yep, I'll ask by video. Thank you very much for the interesting talk, especially for me as a developmental biologist. So I wanted to ask, so you generated this construct as logical sort of circuits, yeah, that you can express and find certain promoters, for example, that are unknown. So I wonder if those constructs can be inducible, and if you try to drive this GXP expression inducible.
Yeah, so one of the nice things about these bacterial DNA binding proteins is that many of them have known small molecule inducers that cause them to change conformation and unbind the DNA. So they're similar to like the TET-R system where, you know, in response to tetracycline or doxycycline, TET-R, which is a bacterial DNA binding protein that's usually bound to DNA, will unbind. They've been engineered to cause them to bind. So
but there's, I guess, some affinity, yeah, of this unbinding type. Because
right.
we in the field we use this estradiol tDNA vectors, for example, that's LHGR4 constructs and so on, and those has been shown to, yeah, for some time to lose the induction, for example. But I wonder if you can control it with your circuit.
Yeah, so we've done a little bit of experimenting by adding different chemicals that are known to affect the proteins that I'm using and their ability to bind DNA. And we do see that when you add the chemicals that the synthetic transcription factors that I've built unbind the DNA, and you can get expression of GFP where it would have otherwise been repressed. They're not super strong at the moment, and I have only validated them in these Nicotiana transient expression assays, so I don't fully know. It would be a good experiment to try applying the chemicals to some of the Arabidopsis that have these circuits encoded and see what happens, but I haven't gotten that far.
And a final question, if I may. So you were inducing a GFP fluorescent protein, I saw, and given that the PIN4 expression is going to be xylem, some protofloem, metafloem cells in the root, which are a part of vascular conductive tissue. So have you considered, is it just GFP, or is like ER-tagged GFP or nuclear-tagged GFP to exclude any mobility of the transcript?
Yeah, these are actually GFP Gus fusions with a nuclear localization signal. So it's a pretty bulky protein that also has this nuclear localization signal that we hope keeps the protein in the plant cell that it was expressed in. But it's a good point.
Yeah. Yes, thank you.
Yeah, thank you.
Great questions. Thank you. Karina has a question.
Hi, yeah, thank you for a wonderful talk. I know it's probably difficult enough to get this started in one plant, but are you thinking about monocots at the moment? And do you know if the same, yeah, if they have the same kind of expression patterns and promoters you can use?
Yeah, so we are thinking about monocots. We had a collaboration to do some transformations in Soteria. It takes a really long time to get those transformants. We don't fully know whether or not these things are functioning well in Soteria. Yeah, where we were actually with that is actually just to try using some RNA-seq data sets to define promoters and then drive expression of fluorescent protein, sort of what I said we shouldn't be doing, but we need to have some idea of what expression patterns we could get to start using as inputs for these synthetic circuits in monocots. I think there's no reason why the synthetic transcriptional activators and repressors wouldn't work in monocots, but I do think that the synthetic promoters might need to be redesigned as I understand it, you need to have, or it's better to have introns in your promoters for expression in monocots, and none of the promoters that I've been using have introns in them. So it's possible that even if the synthetic activator is able to bind in these monocots, they're not going to be good at or able to initiate very strong expression. Something to explore. I think a big barrier for monocots is just time and how long it takes to assay these systems. I mean, we were using Nicotiana as a proxy for Arabidopsis because that's even faster than making transgenic Arabidopsis. And for grasses, we would definitely need to have some sort of quick assay system to cut down on the amount of time it would take. But it's a good goal.
So about these introns, do you just take a coding sequence that usually has this intron in it at the right place, or does this intron have to be inserted into the coding sequence of gene you want to be inserted to be expressed?
So it kind of varies. You can take an intron that's already, like if you're going to express a native plant gene, then you can just clone the whole gene with its introns in it and drive its expression using a synthetic promoter, for example, if you wanted to modify the expression pattern of that gene. If you're introducing something like the synthetic transcription factors, then you would need to take an intron and insert it into that synthetic gene because it, you know, bacterial proteins don't have introns. And that's a little bit challenging. I've had some success taking introns from other genes and inserting them into our synthetic coding sequences. So for example, the GFP that we express in these Nicotiana transient expression assays has an intron inserted into it that I believe is from potato. And I did that because if you don't insert an intron into it, you can actually see agrobacterium fluorescing. So it's able to drive a small amount of GFP expression. And depending on what synthetic promoter you put upstream of that GFP, it will drive more or less GFP expression just because the agrobacterium is maybe not recognizing that promoter in the same way a plant would recognize it, but it's finding sequence in there that it recognizes as a promoter and driving GFP expression. So put that intron in to prevent the agrobacterium from expressing GFP and kind of confounding our results. And it took a little bit of wiggling to find a location in the GFP where we could drop this intron that spliced well, that didn't kind of mess up GFP expression. If that makes sense. It's kind of a long way from what you asked, but.
Okay, well, very interesting. Thank you.
Thanks. So another question from Francisco. Does the asymmetric nature of the biological AND gate, one being input is an activator and the other one is a repressor, result in asymmetry in response to the level of each input? And if so, do you consider this a bug or a feature?
It's a good question. So we're expressing, at least in the Nicotiana assays, the activator and repressor with the same promoter. So hopefully their expression is balanced. But what we haven't done is tag the activator and repressor with any independent fluorophores to really monitor how high the activator and repressor expression levels actually are in any given plant cell. It would be really interesting to try to correlate gate activity with how high the activation or the inputs are, right? So in the absence of that, I don't really have a good handle on how sort of asymmetric expression of these things may be, and asymmetric in terms of the levels of expression, but then, you know, asymmetric in terms of what you're asking, which is the activity of these things. I would love to have more quantitative data about expression levels to try to address how the design of the gate is really affected. And this actually becomes really important when you start trying to hook up native promoters as inputs for these logic gates, because you don't have as much control over a native promoter. So it may be super, super strong. One of your input promoters may be super strong in a specific cell type, and that is driving expression of your activator, but your other promoter may not be as strong and it's driving expression of your repressor. And in the Nicotiana assays, where those two expression levels are balanced, the repressor outweighs the activator and is able to keep your output gene off, right? But in the specific cell type where these things are out of balance, then your repressor may not be expressed strongly enough to turn off your output gene, right? So there's ways in which these things can fail. And if you were to flip, which is the activator and which is the repressor, it may help in cell types where the gates are failing, but I don't have like the reciprocal for all of these things to answer those questions and the quantitative data yet to flesh that out, but it's a good question.
Thanks. Another question just from me is like, what would you see what would you like to see the applications of your research?
I would love to see them in just making crops more robust to environmental challenge. I'd love to see that we can define the relationship between structure and function. So there's a lot of theory in plants about how the structure of even the root system affects the plant's ability to survive in a challenging environment. So like those drought plants that have really steep and deep root systems have been experimentally validated to a certain extent, where we have isolated mutant plant varieties that have these steep and deep root systems, and those are better at surviving in drought conditions. But the problem with the mutations that give you the steep and deep root systems is that it affects expression of regulators that are also normally active in the shoot structure. So it becomes a little bit more challenging to kind of say definitively that the ability to survive in the drought environment is just due to the impact on the root structure. And I'm hoping that by being able to really precisely engineer the structure of the plant, we can say more definitively that we understand the relationship between the structure and function. That the root system change really is the reason the plant is able to survive in a drought environment, for example.
Thank you. While waiting for it, if anyone has any other question, I was wondering, because you have achieved so much, so many brilliant things at such a young age, I was wondering what pieces of advice you would give to an aspiring scientist.
Don't stop. Sometimes science is hard and your experiments don't work, or you don't get your fellowship, or you don't get into the school you wanted to. But I think that being a successful scientist is a lot about perseverance and always trying to push for the science that you find the most exciting and interesting. I think that's where you'll ultimately be the happiest and most successful.
Oh, that's really nice advice. Okay, I have another question from Francisco. Are there examples of complications with using the 5' genomic sequence to identify promoters that would be relevant to prokaryotes as well?
To a certain extent. I think in prokaryotes, one of the things that we do a lot is use too short of a region as the promoter. And there tends to be information, at least for some promoters, that's pretty far upstream of the start site for transcription. So, you can get things like long distance looping and binding sites for transcription factors that affect prokaryotic gene expression that are like way upstream. So in some sense, it's possible that you would need to have more information there. But at the very least, at least in E. coli, I remember from grad school that you want to have at least like 60 bases of promoters that you're capturing sort of the elements of the promoter that interact with the alpha domain of the polymerase. This is more detailed than you needed. Yes. Sort of the short answer to your question.
All right, that was very good. Yeah, so I'll be wrapping up this Q&A session now. Thank you to everyone in the audience who has asked a question, and thank you especially to you, Jen, for answering all the questions so well, and yeah, just for giving us time to give us this amazing presentation and share us your work. Yeah, we wish you all the very, very, very best with starting out your new lab. We're very, very excited for you. So yeah, our next webinar will be next Wednesday, and it will be on bioeconomy. It will be delivered by Dr. James Phil, a policy analyst specialized in industrial biotechnology and synthetic biology. We can register through Eventbrite to join us, and we'll send you the link in a follow-up email. So yeah, we're going to end this meeting now, and we really hope to see you in future events. Thank you so much again, Jen.
Awesome. Thank you so much for having me. Bye, everybody.
It was a pleasure. Bye-bye.